Hello, everyone, and welcome to today's webinar titled Clinical Performance Characteristics of a Molecular IVD-cleared Multiplex Respiratory Panel for SARS-CoV-2, Influenza A, Influenza B, and R. This webinar is a part of the 13th Annual Precision Medicine, Genomics, Genetics, and Molecular Diagnostics virtual event series. I'm Melanie Chan of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Dr. Harita Veereshlingam, Clinical Staff Scientist at Thermo Fisher Scientific. Dr. Harita has over 17 years' experience in the life sciences industry. She has a Ph.D. in molecular biology at the University of North Texas and a master's of microbiology at the University of Mumbai. Some of her professional achievements include leading numerous clinical validation studies for IVD devices, including 510K, IVDR, and CDSCO submissions. She specializes in clinical trials, regulatory compliance, and program management, and has the dedication in advancing reliable molecular diagnostic products to improve diagnosis and treatment timelines. For a complete biography on Dr. Harita, please visit the Presenter tab from the menu on the left of your screen. Dr. Harita, you may now begin your presentation.
Melanie, thank you so much for that wonderful introduction. I'm really happy to join you all to talk about our newest offering in the genetic testing solutions space. This is the TACPath COVID-19 Flu A, Flu B, RSV Select panel. It's a 4-in-1 viral testing solution, which has been designed to help you manage routine handling, seasonal and outbreak-related testing. It is a FDA-approved 510K IVD kit. It's a multiplex PCR diagnostic solution and helps you differentiate and detect 4 very commonly found respiratory viruses. In addition to detect the co-infections so that you can be confident with your results. All right, so let's take a look at what this kit entails, right? So as I mentioned, this is a clinically validated IVD kit so that you can be confident with the results you get. It's a 4-in-1 diagnostic kit, helps you detect and differentiate 4 different respiratory viruses from a single patient sample. In addition to the 4 viruses that you detect, the kit also helps you to have more confidence in your data because it has an internal RNA-seq control, which ensures that you have collected the sample correctly. The integrity of sample is maintained. It goes through extraction and detection, so you have high confidence in the data. And finally, it's built for diagnostics, right? So it's validated with the diagnostic instruments and software. So you can leave the validation part to us. We've already done that for you. All right, so briefly going over what the 510K clearance entails, right? So this is a very, very high level overview of what it means to get a IVD kit 510K cleared by the FDA. The 510K path requires you to demonstrate substantial equivalence to an existing device, which is called as a predicate device. The existing device should be a device that has been cleared by the FDA either via the de novo process or via the 510K process. The investigational device, in this case, I'm referring to the TACPath device as the investigational device, should show should be compared to the predicate device with respect to technological characteristics, with respect to the intended use, and above all, there needs to be performance data that should show, again, substantial equivalence to the predicate device. This data includes analytical tests, it includes clinical validation, and a whole slurry of data that is presented to the FDA. Since we are going to talk a little bit more about the clinical validation study in this talk, I thought I'll put in a little blurb about what the requirements for this clinical validation study was. So besides the fact that we had to do a clinical validation study to show substantial equivalence to the predicate device, the study had to be a prospective clinical study, which was done in the United States, in representing different geographical regions. So samples were collected from many different regions within the United States, and was conducted through a complete flu season, with a minimum of 50 positive clinical samples for SARS-CoV-2 and flu A, and a minimum of 30 positive clinical samples for flu B and RSV. So that was, in some ways, the design of the study at a very high level. All right. So now coming to the intended use. So now, as most of you are familiar, in any IVD device, intended use is very, very important. So for the TACPath COVID-19 flu A, flu B, RSV select panel, the intended use, I'm not going to read through the entire thing, but again, a gist of this is that this is a multiplex real-time reverse polymerase chain reaction in vitro diagnostic test for the qualitative detection and differentiation of SARS-CoV-2, flu A, flu B, and RSV. Nucleic acids are isolated and purified from nasopharyngeal and or anterior nasal swab specimens obtained from subjects suspected of respiratory tract infection, right? So there's a subject who show signs and symptoms. That's what the intended use of this device is. Clinical signs and symptoms of respiratory tract infection due to either of the four viruses can be very similar, and hence differentiation between the four viruses um, is very beneficial for a clinician. A note here that the device detects both RSV A and B, but doesn't differentiate between RSV A and B, right? So, and we'll see how that works in the few slides. So I'm going to jump to the next slide and just making a note here that the intended use must be followed to comply with the 510K regulations that are set by FDA. So that's very critical. All right, so moving on to the kit itself. So the TACPath COVID-19 flu A, flu B, RSV select panel is a multiplex real-time PCR diagnostic kit which targets SARS-CoV-2, flu A, flu B, and RSV. And all of those four are not only detected, but they're differentiated as well. There are two samples that have been validated with this kit. That's the nasopharyngeal swabs and anterior nasal swabs. The kit comes in two formats, a 200 reaction format and a 1000 reaction format. So it gives the user a little flexibility on what they want to use. And the assays targets, as I mentioned, 4 different viruses, right? So here we go a little deeper into what are the genes that we target. So you'll see that the assays have been designed to very conserved regions within each of the 4 viruses. So for SARS-CoV-2, we target ORF1A, 1B, and N genes. For influenza A, we target PP1 and M genes. For influenza B, M and NS genes are targeted. And for RSV, there are 3 targets, NP, M, and L protein genes. In addition to the 4 viral targets, there is an assay that targets RNA-seq, but this is the internal human sample collection control. It ensures that samples are collected and they are present in your sample, right? So that negative samples, negative data especially, is truly negative because you have the presence of RNA-seq. All right. So let's move on to the workflow, right? So again, I'm speaking to a very high level overview of the workflow. As I mentioned earlier, this is a IVD kit, and the entire workflow from from the time the sample is received to the results output, the entire workflow is validated as required by IVD regulations, and it is automated, right? So that makes it a lot easier for testing using this kit. Sample extraction happens on the Kingfisher Apex DX purification instrument using Magmax DX viral pathogen nucleic acid isolation kit, followed by PCR on the QuantStudio 5DX real-time PCR system. And data analysis happens on, again, an IVD software, which is the Diomni, applied by Systems Diomni software. This software not only helps in analysis of the data and provides results in the format that's easily readable, it also helps you connect to the two instruments and hence in the workflow, right? So streamlined workflow with minimum hands-on time. All right, so let's take a look at what the kit comes in. So when you purchase the TACPAT COVID-19 Flu A Flu B RSV Select panel, what comes in the box is the assay, right? So the assay which targets the 4 viruses as well as RNA-seq. You also get a positive control. Now this is actually very, very important to ensure that you know, a positive is a positive and a negative is a negative, so you have control over your data. The positive control comes with are actually targets for the 4 viruses. So SARS-CoV-2, Flu A, Flu B, and RSV targets are present in the positive control. The negative control has only the RNA-seq target. And along with the assays and the positive control in the kit is included the TACPAT one-step select master mix, no rocks, right? All right, so here is a snapshot of what the plate looks like when you set up your reaction. Now note that this is a 96 well plate. 96 well plate goes into the Kingfisher instrument and the subsequent PCR instrument, right? So you can set it up in 96 wells, which means that if you are allocating 1 well for positive control and 1 well for negative control, and that is a must, unless a positive control and a negative control are on each single run, the run is invalid. Right, so you have to allocate 2 wells for the controls. Now you're left with 94 wells in which samples can be added, which means in one single run, you can test as many as 94 samples. Do you need to have 94 wells, 94 samples? No, you can test as little as 1 sample, right? So it gives the end user a lot of flexibility on how many samples they can test in one go. Very very helpful when you have different number of samples coming into your reference lab. All right, so what does the workflow looks like? So you get samples. These are, as I mentioned, NP swabs or AN swabs that come in. And we have some recommendations on what could be the sample collection media, followed by sample extraction, again automated on the Kingfisher Apex DX purification system using the Magmax DX viral pathogen nucleic acid isolation kit, followed by qPCR runs on the QuantStudio 5DX real-time PCR instrument, and results interpreted, and a report is generated using the Diamni software. All right. Now, so that was the workflow, where, streamlined workflow, minimal hands-on time. With that, I will move on to looking at some of the data that we were able to generate during the clinical validation study for 510(k) requirements. Okay, so the objective of the study was to evaluate the performance of the TACPAT COVID-19 flu B RSV select panel in both specimens, right? The NP swab and the AN swab. And we used a comparator approach in which clinical calls for each of the 4 viruses in the 2 different specimen types was compared to clinical calls in the 2 different sample types using a comparator device. And any sample that produced a discordant result was further tested with a resolver device. The resolver device was also a 510(k), was also a FDA approved device. With the same intended use as that of the TACPAT COVID-19 flu B RSV select panel intended use. And the data from the discordant testing, however, was not used for agreement calculations, and you will see that in the subsequent slides. We'll jump into the data. All right, so here is a table that shows the sample demographics of samples that were collected during our prospective sample collection between February 2023 and February 2024. So it actually spans 2 flu seasons, part of 2023 and most of 2024. And what did we see? I don't expect you to go through the sample demographics here, but here is what we got, right? During that prospective sample collection, we collected specimens from 1,840 symptomatic individuals, and 1 NP and 1 AN specimen was collected from each of the individuals. Samples were collected in UVT, and the collection order, because we were collecting 2 specimens from each subject, the specimen collection order was swapped. So first NP and then AN, and then the next subject we collected AN, then NP. This was to reduce any bias that might creep in, because specimens were collected from the same subject, and we actually did not see any bias in which specimen was collected first. Specimens are collected from 14 different collection sites that represented geographically diverse regions within the United States. And after exclusion of the samples, either due to protocol deviations at the collection site or the testing site, we ended up with 1,620 NP swabs and 1,541 AN swabs, which were used in the analysis. And in a moment, we'll look at what the data was. All right, here. So these are 2x2 tables showing concordance of the clinical calls between TACPAT COVID-19 flu B RSV select panel and the comparator device for each of the 4 targets. So that's why you see 4 different 2x2 tables, and this is of course only for NP swabs. We'll look at AN swabs in the next slide. All right, so what do we do with a lot of numbers here? What I want you to focus on is the PPA NPA, which is positive percent agreement and negative percent agreement for each of the 4 targets. You will see that we had very concordance of data. More than 90% concordance for all 4 targets in NP specimens, right? So that's actually very, very good concordance. All right. So similarly, these are the 2 by 2 tables for AN swabs, and again, we see very high concordance, greater than, I think this was all 95%, and higher concordance to the competitor device. Um, do notice that at the foot there's a footnote on each table, each of the 2 by 2 tables. This footnote shows the data from discordant testing. Remember, I mentioned that when there were any samples that showed a discordant data between investigation and competitor device, those samples were tested with the third device, which was a 510K clear device again, and data from that is presented in footnotes. What we found was that there was nothing blaring, right? So it was as is expected in samples which are at or below LOD. You see the data could shift either ways, right? So if it is called positive by one, it could be called negative by the other instrument, and so on and so forth. So that was a trend we saw across the board for these samples. All right, so the next set of data is data from samples that were collected during the enrichment phase. So what was enrichment phase? Because this was such a large study that went for over a year, there, during the off time, when off time, when I say off time, I mean that when flu, it was not flu season, and the number of positive samples decreased significantly. um, we did an enrichment sample collection in which we were able to collect from samples we knew had one of the four viruses as a causative agent, right? So eligible subject with a positive PCR test no more than 3 days prior to enrollment were included in the study. And this enrichment sample cohort only happened after meeting a caveat, which was that the first 1,000 subject during prospective collection were collected, and the minimum number of SARS-CoV-2 positive samples were collected. Right, so we're done collecting the minimum 50 SARS-CoV-2 positive samples, and a total of 1,000 subjects were enrolled in prospective collection. Once that was done, then enrichment sample began, and this was only uh, established at 3 of the 14 different collection sites. And what did we find? We found that of the 69 samples that were collected for NP, we again, concordance with the comparator device, we saw a PPA of 97.9% for flu A, 92.3% for flu B, and 100% for RSV. Again, very, very high PPA. And you can see the NPA numbers also very high, greater than 95% for all 3 targets. As I had mentioned, there were no SARS-CoV-2 samples collected during the enrichment phase because we had already met our numbers for SARS-CoV-2. All right, so that was NP, and like in prospective sample collection, both NP and AN specimens were collected from all subjects, and you can see that again here we had very high concordance to the comparator device, 100% PPA for flu A, 92.3% for flu B, and 100% for RSV. So this data, as can be seen from the numbers, shows that in a clinical study, we found that the TACPath COVID-19 flu A, flu B, RSV select panel had very high concordance to the comparator device, and both in prospective and enrichment clinical sample cohorts, right? So, what were the key takeaways? Well, from both the analytical, which data was not really presented here, but is available, we saw very high sensitivity and specificity of the TACPath COVID-19 flu A, flu B, RSV select panel to the comparator predicate devices. We also saw that this select panel helps you to simultaneously identify 4 major respiratory viruses, right, from a single sample. So highly efficient, um, easy to use. It's a user-friendly workflow, as I have described. It's compatible with established LIMS systems and protocols, and allows seamless integration into existing workflows. There's using this, as you can see, there as many as 94 different samples can be tested in a single time. So it's highly efficient, saves time and resources in a clinical setting. And finally, because it detects such critical targets from a single specimen, it is of high clinical relevance, offers critical information for patient management and diagnosis. So with that, I will take any questions that might be coming our way.
Thank you, Dr. Harita, for your informative presentation. We will now start the Q&A portion of the webinar. We have lots of questions that have already come in, but if you haven't already submitted any and would like to, please do so now in the Ask a Question box located at the bottom of your screen. Now, let's get started. Our first question asks, what are the benefits of selecting an IVD assay versus an RUO that is validated by the testing lab?
Ah, so Melanie, I think uh, the as I had mentioned, to the rigorous testing that has happened on our end, right, to get IVD clearance, to get 510(k) approval from FDA, we've done a lot of validation on our end, which automatically means that the customer is getting a validated product. They don't have to do any validation on their end. They can have greater confidence in the data they get, and the onus is then on us to provide the data that gives them confidence in the product.
Great, great. The second question that came in asks, why is RNA SEP so important when testing for respiratory infections?
So RNAsP. Okay, so RNAsP, is an internal control. It is one of the genes that is always present. It's present in all our cells, right? So presence of RNAsP in a sample ensures that the sample was collected. Right, so there's actual sample. It's not like sometimes the sample collection may not happen nicely. It may people don't take either the NP or AN swabs correctly. Not enough cells are taken. So if RNA-sp is not present in your sample, a negative result especially could mean either that there was no sample or that the subject is truly negative. So presence of RNA-sp on the other hand gives you confidence that the sample was definitely present, and a negative means that there is no target virus present in the sample.
Thank you, Dr. Harita. The next question asks, is H5N1 included in the pool of flu A for the TACPath IVD? If so, what is recommended when there is a flu A positive?
Okay, that's a very good question, especially in this this time where there's definitely and more and more cases of H5N1 infections. Yes, our flu A assay does detect, so it does have coverage for H5N1. However, it doesn't differentiate, right, between H5N1 and the other flu viruses. So the recommendation would be that if a subject has flu A positive, right, then and a physician or the clinician thinks it's necessary for them to know whether that is due to H5N1, they would have to do a follow-up test, which with a H5N1 specific assay to confirm whether or not they have influenza because of H5N1.
Okay, great. And it looks like we have time for one last question, and that asks, how does the TACPath IVD work when there are mutations and new variants of SARS-CoV-2, influenza, and RSV?
Oh, that's a very good question. So there are a couple of things that are happening, right? So first, as I had mentioned, our assays are designed for highly conserved regions, right? So we know that the regions that our assay designs are highly conserved, very unlikely that they will not pick up the mutations that are in circulation. Having said that, because this is an IVD kit, because this is approved by the FDA, we as manufacturers are required to do to constantly keep track of what are current mutations in circulation and ensure that our assay detects those. If we do not, or if there is a mutation, say in the next 1 year, or through the course of the product, that we know doesn't affects the detection of our assay, then we are required to make that information available to our customers and to the FDA, so corrective measures can be kept in place. All right. Also, as I mentioned, the fact that each of our assays targets more than one gene, there are less chances of that happening. But yeah, we are constantly doing post-market analysis. We're constantly keeping track of what mutations are in circulation to ensure that our assay detects those.
Great. Thank you, Dr. Harita. Do you have any final comments for our audience today?
Well, first of all, thank you, Melanie, for giving me this opportunity to talk about the product. We are excited. I think this is a very, very robust product, and we look forward to hearing from our customers on how they find the workflow, how they're able to use it. We're looking forward to it. It's exciting. A lot of work has gone into it, and we hope it'll meet the needs of our customers.
Great. Thank you again, Dr. Harita Veereshlingam, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. We would also like to direct your attention to their booth in the exhibit hall after this webinar for any resources and any additional information you would like to see on them. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until May 14th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care and goodbye.